Okay, so thanks everyone for joining the EV Club today. So my name is Clotilde Théry. I'm the immediate past president of ISEV. I quit my functions at the last ISEV 2022 General Assembly. And I'm chairing today the EV Club. So the EV Club was originally initiated by Ken Whitwer during the pandemic, and it has now become an ISEV event, and we are sharing the organization with Ken and Metka Lenassi, who's now the chair of Science and Meetings. So this week we are very happy to have Gisela D'Angelo joining us to present her work, her paper, recently published paper, which will describe, well, not exosomes for once, that's good. That's going to be another type of vesicle, and in a very interesting model, which is the Drosophila wing imaginal disc development. So Gisela, maybe you can start sharing your slides.
Well, thank you, Clotilde, for asking me to present this recent data that we obtained a few months ago, and I will be ready to, if you want to interrupt, but I think you prefer to go through the whole presentation. So let's see.
I think it's better for a Zoom presentation to not interrupt. So you give your whole presentation, and then we'll allow the discussion and reactivate the microphones of all participants. So thank you.
Thank you. So I'm going to talk about, as Clotilde was saying, about extracellular vesicles as mediated of intercellular communication. And I will talk essentially about development, how they interact and play roles in development. But I would like just to finish before the end of the talk to give you a flavor of how these vesicles are also involved in cancer progression. So I guess that you are all used to these extracellular vesicles, but nevertheless, I will go through a little introduction, if I can do that. Okay, thank you. So you know that the intercellular communication, or the transfer of information between two cells, is essential for the body organism and for the body organism generation and for the homeostasis. So there are multiple ways by which the cells can contact each other and transmit information. This could be, are you seeing my pointer? I don't think so. Well, okay. So they can contact each other by direct contacts, or they can also release soluble factors that from the sending cells will act on the receiving cells. But there is now other different models of intercellular communication, such as autocrine, where the cells release the factor that will react on itself, paracrine, where the cell will send some factor.
Sorry, Gisela, I see a screen, the screen of your pointer on the screen. Oh, no, sorry, maybe it was me. Sorry about that. Go ahead.
What should I do? Do you see my pointer?
Now we see your pointer. Maybe you could make it as a red arrow, but maybe it's not necessary. You can leave it like that. It's okay. Sorry for interrupting.
Okay.
Yeah.
No, it's okay.
No, go ahead. We see the arrow.
Okay.
Yeah.
Oh, I'm sorry. So there is also, as I was saying, another mechanism, which is called the paracrine mechanism, by which the cells, the sending cells, will release a factor, and this factor is going to be soluble and is going to act on the recipient cells. And endocrine, where the factors are released inside the bloodstream, like the hormones. And finally, the recent mechanism, very novel mechanism that we are all used to now, is the EV-mediated communication between the cells in the whole body. So this is actually the way I call now, it's EV crine system for cell-cell communication. And as you know, when you have a secreting cells, and mainly all cells in the organism, they can secrete extracellular vesicles. And this could be achieved either by MVB fusing with the plasma membrane and release the exosomes, or by a budding process at the plasma membrane, which will end up by releasing ectosomes. Whatever, these two types of extracellular vesicles contain genetic materials, proteins, lipids, and other things that could be, by being secreted by the sender cells, act on the recipient cells to modulate and to reprogram the fate of the recipient cells. So although much is known, as Clotilde actually pointed out, that in general people talk about exosomes, today I would like to pinpoint that exosomes are really different vesicles, and much less is known about the origin and the physiological relevance of this type of extracellular vesicles that bud at the plasma membrane. So to do so, we will focus on the plasma membrane, and in particular, I decided to investigate much more closely the epithelial cells, which are polarized cells and contain an apical domain, which is subdivided in between planar regions, shown here, and the protrusions that in the epithelial cells are termed microvilli. And the particular of this epithelium is that they have specific lipid domains in these microvilli that are extremely enriched in cholesterol, here showing in red, and you have other lipids and myosins and also actin bundling proteins to maintain the structure itself. But more importantly, what we know now is that there is a specific microvillar protein that is called Prominint or CD133. So this protein tightly associated to cholesterol in this microvilli domain, and from there, it was showing that it's able to incorporate in this cholesterol-enriched domain, and is also considered today as a stem cell and a cancer stem cell marker. And recently, I mean, a few years ago, it was showing that this microvilli, when the prominint was overexpressed in the MDCK cells, we were able to see a little immunostaining here, electron microscopy here with prominint. And more recently, 15 years ago, in CACO cells, the epithelial cells, we can see that these vesicles, these microvilli, release vesicles here that are supposedly positive for prominint. But so far, we know now that it's a signature of extracellular vesicles that are released into the extracellular space. However, at that time, when Corbeil and Maresco showed that these vesicles were occurrent in epithelial cells, no function was attributed to them. So to tackle this question and try to understand whether there is a relationship between microvilli, extracellular vesicles, biogenesis, and a potential contribution of these particular EVs in mediated intercellular communication in development, I focus on the Drosophila wing imaginal disc as a model system and using the morphogen Hedgehog as a readout. So in particular, the wing imaginal disc is composed of columnar cells, which carry and are decorated with microvilli. And as I told you, they are epithelial cells, so they are polarized with an apical and basal domain. And in the wing pouch itself, this region here that is delimited by the circle gray here in the wing imaginal disc, the schematic of the wing imaginal disc, they carry epithelial cells which has a columnar shape, whereas the epithelial columnar cells are separated from the squamous epithelium by a lumen. And it's within this lumen that the vesicles could appear. And as you can see here, this is a ultrastructural analysis of the wing imaginal disc, a wild-type disc of Drosophila, where we can appreciate here the squamous epithelium that is much more enlarged, and the columnar epithelium that is much more horizontal. And what you can observe is that within the lumen, you can see these membrane vesicles that are found at the close proximity of the microvilli. And when we enlarge a little bit, we zoom the image, we can see the round shape of them. And we evaluate the size and the distribution of these vesicles altogether by electron microscopy, doing electron microscopy, and you can see that here in the columnar epithelium, you do observe multivesicular bodies and also the vesicles nearby the microvilli. But the size of these vesicles from inside the MVB, which are later will be the exosomes, are different as the ones we observe here in the lumen. The size distribution doesn't really argue that these vesicles that are at close proximity of the microvilli are extracellular vesicles that probably can arise from budding of their plasma membrane. But to carry out that and to closely look at that, we carry out a lot of electron microscopy during cellular sectioning. And what we found is that we can observe at the microvilli some budding events. And these budding events are really close at the microvilli, again, to these extracellular vesicles, or membrane vesicles. Then by doing electron tomography, we can see here that in red, we were delineating these extracellular vesicles or membranous vesicles in the lumen. And by carrying here the electron microscopy, the electron tomography and the film, we can see that these structures are indeed attached to the microvilli, but they go on and they are released in the luminal space. Okay, so next, to make sure that Prominint was indeed expressed and distributed to the microvilli in the wing imaginal disc, we generate an antibody, especially in the Drosophila, and we found that Prominint distributes very above the epithelium, that is the apical marker, and disc large, a subapical marker in the wing imaginal disc epithelium in Drosophila. And you can see that the Prominint here in red shows the distribution of dot-like structure, distribution or staining, that it's indicative of microvilli, as we could confirm by looking at the ultrastructural analysis. Now, I don't know if you are familiar with the flies, but we can do a lot of tricks and genetic applications with, and we can target a specific expression of a protein in one of the compartments of the wing imaginal disc. So the wing imaginal disc, as shown in here, is divided in ventral and dorsal for this particular image, and we can express apterus, that it's a marker of the dorsal compartment. And in this particular case, if we target the expression of prominint RNAi in the dorsal compartment, we could use the ventral compartment as an internal control, because the ventral compartment will be wild type. And when we do so, and when we knock down prominint RNAi, a prominint expression in the dorsal compartment, you can see a disappearance or a fade of the staining of prominint-like in the dorsal compartment showing here, whereas in the ventral, which corresponds to the wild type compartment, you can see these dot-like structures here staining, as I was showing in the wild type disc, because this is exactly the same than the wild type, the entire wild type disc. Now, if you use also this antibody in a prominint mutant that we generate, or in a Cas9 mutant, you can see that the staining of prominint is also degraded. So this suggests and indicates that prominint is not only present in microvilli, in wing imaginal disc, but it's also expressed in the microvilli, and apparently it contributes to the structure of the microvilli. And here is only to show you the decrease of the prominint intensity when we knock down the protein. So now, to make sure that prominint is critical for microvilli integrity, what we did is we knocked down prominint now in the posterior compartment, also another compartment of the wing imaginal disc. And by doing so, we will compare the posterior, which will be the mutant compartment, with the anterior, which will be the wild type compartment. And by doing so, we can see that in the posterior compartment, here you have the lumen, the squamous epithelium, and the columnar epithelium. Here is the AP border, anterior posterior border. You can see that the structure, the microvilli structure that is harbored by the anterior compartment, is completely abolished here, or at least strongly affected. And we will measure the length of the protrusions of this particular disc where prominint was depleted. We can see a strong reduction of the protrusions themselves, arguing again that prominint is critical for the microvilli integrity in the wing imaginal disc. So to make sure that it was not an artifact or something that was wrong with the experiments or whatsoever, we also use and knock down proteins that are involved in the microvilli structures. As I told you, these microvilli are very enriched in cholesterol, and the lipid asymmetry of the plasma membrane plays a fundamental role in all the protrusions, in the makeup of protrusions. So to do so, we use ATP8B RNAi. ATP8B is a flippase enzyme that is going to flip some lipids from the outer to the inner, membrane at the microvilli. And we use also quail RNAi, which is an actin bundling protein in Drosophila, is the equivalent of villi. And in both cases, we can see, again, by expressing in the posterior compartment, either ATP8B or quail RNAi, that the structure of the microvilli is reduced or amorphic, like in here. And again, the pattern of the protein like expression in this wing imaginal disc as compared with this wild type doesn't show again the dot-like structures here in the ATP8 posterior compartment, or here where you can see it's a posterior compartment of the quail RNAi, where the prominent was abolished as compared, expression was abolished as compared to the wild type. So this altogether suggests that the length of microvilli are affected in both conditions, and this is correlated with an absence of extracellular vesicles that we could observe in the previous slide in the wild type wing imaginal disc, and that now they are absent not only in prominent-like RNAi, but also in ATP8 and quail depleted disc or depleted compartment. What I could say up to now is that microvilli are the site of EV biogenesis, as we observe, that the biogenesis rely on an active interplay between prominent actin cross-linking proteins and microvilli lipids. But now the main question that we reveal the origin of these microvilli-derived extracellular vesicles that are indeed different from exosomes is whether they could play a role in the long-distance cell-cell signaling in development. And as I told you, I was using Hedgehog, the morphogene Hedgehog, as a readout, And so Hedgehog plays a fundamental role, not only in development, but also in diseases. And the way it contributes to that is that Hedgehog, as all morphogenes, is expressed by source cells and then is released by these cells in a gradient-dependent manner, in a manner that then the cells that are close to the source of Hedgehog production will receive the intense signal, intense protein. And as long as we go, as far as we get away from the source, the distance increases, but also the morphogene concentration decreases. And Hedgehog does that by displaying its gradient of morphogenic action in the extracellular space. And then Hedgehog is fundamental for embryonic development, to mention few, cellular growth and differentiation, and also for stem cell homeostasis. But what is very important to know is that Hedgehog in loss of gain of function generates a lot of defects. Like here, if you look at this embryo, the absence of Hedgehog reduces the size. This is in a wild type mouse embryo. Here in the Drosophila, the larval structure here, the larval structure of the wild type larva, in the absence of Hedgehog is reduced, and the patterning of the larvae is totally dysregulated. In humans, a loss of sonic Hedgehog, the homologue of Hedgehog in mammals and humans, could generate additional or loss of fingers, for instance, digits. And in cancer, it's one of the key factors when dysregulated that it's involved in basal cell carcinoma and of many different types of epithelial cancers. So knowing that, we reason that probably microvilli-derived extracellular vesicles could carry Hedgehog to develop his its long-range activity, and we focus on the developmental stage and actions of Hedgehog. So in the wing imaginal disc, the source cells that express Hedgehog are the posterior. Here you have a wing imaginal disc. As I show you, this is the wing pouch, the circle here. And you have divisions, a division between different compartments. And in the posterior compartment, which are the source cells that produce Hedgehog, Hedgehog is made here, generate here, and is released and act on the anterior cells and activate under a gradient form different target genes, such as engrail, patch, which are high and medium level of Hedgehog, meaning that they will receive either a lot of Hedgehog or roughly half of the concentration, and far away from the source of Hedgehog, the posterior compartment. Within the anterior compartment here, we will see the increase of DPP, that it's a long target Hedgehog gene. And what we had shown in the last year is that Hedgehog long-range activity, meaning for Hedgehog to be released from posterior cells to act on very far away in anterior cells, 30 microns away here, this is the distance. Hedgehog can be released by two different poles of the apical epithelium, either by the basal. I'm not sure that you can see everything because I have my band here. Can you? Okay, well, so Hedgehog will be basally released and activate, in that case, engrail and patch, the high and medium target genes, or when Hedgehog is released by the apical, it will display and will be able to deploy its long-range activity. So the question we wanted to ask is whether or not Hedgehog, by being released at the apical plasma membrane, reaches the microvilli, and from there, upon a prominent effect, will be released on extracellular vesicles. That's the question we would like to answer. And to do so, we hypothesize that Hedgehog reaches this particular structure, and we knock down prominent in the wing disc. We analyze prominent Hedgehog distribution in the wing disc, imaginal disc. And as you can see here in the wild type disc, you can see that prominent and Hedgehog are found at the microvilli level, and sometimes, not always, they colocalize. And the reason we don't see a full localization of both proteins is that the squamous epithelium, as I showed you previously, is not aligned like in the gut, for instance. The cells are intercalated in between them, and you can see different microvilli when we do a scanning electron microscopy that are pointing up in the wing epithelium and the apical surface. But when we knock down prominent in the posterior compartment, we observe here that the staining and the colocalization of Hedgehog and prominent is totally abolished. And this, as related to what I was showing you before, is because we don't have any more microvilli in the absence of prominent. So in addition, we also check that in the wing imaginal disc with wild type or deprived of prominent, the distribution of Hedgehog itself is not the same. In this wild type disc, you can see that Hedgehog distributes above the subapical marker cadere, whereas when Hedgehog prominent is knocked down, Hedgehog distributes, is not located above the cadere expression level, but underneath, suggesting that in any cases, Hedgehog, by the absence of microvilli, the absence of prominent, cannot reach the structures. So this led us to ask the question whether Hedgehog also, in the absence of prominent, could contribute to the long-range signaling. And as I told you, Hedgehog in the wing imaginal disc expressed in the posterior compartment acts on the anterior compartment and increases its target gene expression. And grail, where you have two cells in the anterior compartment, patch, I'm sorry, it's a little bit misplaced, patch that activates 4 cells in the anterior compartment, and DPP in the anterior compartment, the range of DPP expression goes from 8 to 12 cells. And this is the normal configuration that we can see in a wild type disc. However, when we knock down prominent, we can observe that Engrained expression is not affected. We again maintain the 2 cells in the anterior compartment. Patch again, we can see the 4 cells, but in contrast, the expression of DPP target genes is completely reduced and by above 50%. And this correlates with the absence of microvilli, the absence of hedgehog at the apical membrane, and with the necessity of hedgehog to reach these apical structures to be released and display its long-range signaling. So now, the same happens when we deplete quail. As I told you, quail, it's a actin bundling protein. When we knock down quail in the dorsal compartment, we don't see the expression of prominent in the anterior compartment, showing in the posterior compartment here. I'm sorry, in the ventral compartment, we have prominent in the dorsal. It's absent, as you can observe here. And simultaneously with that, we also decrease DPP expression in this particular genetic condition. However, when we overexpress prominent by using a Prominint-GFP protein, we can see that prominent really is enriched at the microvilli level, and that DPP expression is totally enlarged as compared to the wild type, as shown in here. So we reason that if this happens, is because probably the overexpression of Prominint-GFP will increase the number of extracellular vesicles within the wing imaginal disc. And as we can see here, and with this close-up, we can see that the microvilli are present, but they are shorter than in the wild type compartment. And this is probably because they release so many extracellular vesicles that the structures doesn't have enough time to be resynthesized and reproduced. And if you have a close look on these structures, you can see the microvilli shorter here, the microvesicles, the ectosomes or vesicles nearby, and the size, again, that is much more important than the size of the ILBs. In addition, what we can say is that the number of vesicles in this particular genetic background is also increased as compared to wild type conditions. Based on what we observe, we can now think that probably prominent and hedgehog travel long distance associated to one another. But before looking at that, we carry out some immuno-EM analysis here in the wing imaginal disc, where we overexpress hedgehog with RFP and Prominint-GFP, and we can see that the beads here positive for hedgehog, and the biggest one is prominent, the smallest one is hedgehog. You can see that both structures localize to microvilli in this wing imaginal disc. And when we look at the vesicles itself, you can see that the vesicles carry both proteins here in this immuno-EM analysis. Now, the overexpression of prominent and GFP in the wing imaginal disc increases the size of the disc. And you can see here, you can see this enlargement here, where you can see cell mask is to mark the membranes of the cells, RFP for hedgehog and GFP for prominent. You can see these little dots that are almost white, meaning that they have the 3 colors together, the 3 proteins together, as you can depict it here. And although we can see some structures that are only carrying hedgehog here, like red and blue here, which are depicted here, they only represent 20% of the 80% of the total vesicles that carry both proteins. And if I show you now one of these little dots, you can see that they move along the extracellular space in the wing imaginal disc associated to one another. So with that, I think that I can show you that microvilli-derived extracellular vesicles are a means to exchange signals during development, that prominent is a key determinant of microvilli formation in the wing disc epithelium, that microvilli integrity is also dependent and critical for the biogenesis of extracellular vesicles, and that these microvilli are the preferential site for the biogenesis of the disease, and then finally that these microvilli-derived extracellular vesicles ferry hedgehog across the tissue to display hedgehog long-range signaling. And before I finish, I would like just to give you a flavor that the role of this prominent CD123 also in cancer, And they are also involved, cancer cells and breast cancer cells when they start to invade and to generate invasion and metastasis. One of the mechanisms they use not only is extracellular EMT transition and invasion and migration, but they also promote angiogenesis to allow the cancer cells to enter the circulation and to be able to metastasize far away and to create a metastasis niche to prepare the organ for the organotropism. So what happened is that these cancer cells, and mainly the triple negative breast cancer cells, they are enriched in CD123 or Prominint, and we were wondering whether these CD123 positive vesicles were able to play roles in angiogenesis, EMT, on pre-metastatic niche formation, among others. And we had now isolated from this triple cancer negative, triple breast cancer cells, the 468 cells, the small and large vesicles as shown here and here in the Western blot. And what Mireia was able to show a student in the lab is that these vesicles, once applied on endothelial cells, UVEC cells, the human endothelial vein umbilical cells, seated on matrigel, she can see that the addition of extracellular vesicles, either small or large, they are able to form these cords that resemble, and they are capillary-like structures. And this is not important only because they are able to do that, but the property that these vesicles have is above the one of the specific endothelial growth factor, the VEGF. And with that, I would like to thank you, the people in the lab. This is the team in the lab. And my collaborators for this work that are Renata Basto and Sarah Watson and Rafael Rodriguez for the last part, and Anne-Sophie Massé, Lucie Sengeminov, Laurent Ruel, and Pascal Théry. And with that, I would like to thank you for your attention. I am ready to take some questions, if any.
Well, thank you very much, Gisela, for this beautiful presentation, this amazing electron microscopy pictures. Sorry, I have to just maybe reactivate the sound of people. I hope I did what's possible, but anyway, I'm not so familiar with this anyway. But maybe I'll start with a few questions. So yeah, again, your electron microscopy images are really amazing, and it's very important to have this kind of images for when we analyze EVs, because indeed you were very careful to try to, to determine whether your EVs were coming from the MVBs or from the plasma membrane, and convincingly showed that these Microvilli-derived EVs are abundant and useful actually to do something. So I think I hope it convinced people listening that it's always important to analyze not only the small EVs and what you think is exosomes, but also the others. So yeah, I was going to start asking questions, but now I see Kai has a question. Can you unmute yourself? Can you speak, or did I not do?
Sure.
Yeah, you can. Okay, good.
So, so the Hedgehog has the palm dilation site in the N terminal. I believe in the N terminal, and in the C terminal has the another modification. So are you able to identify this modification, how important for the Hedgehog to incorporate into these vesicles?
Yeah, hi. Yeah, as you mentioned, and I think that I show, Hedgehog is the only molecule in the whole animal system that is dually modified. And Hedgehog, because of that, Hedgehog cannot be released by the cells. So it needs to be somehow, the lipid adducts needs to be shielded. So the only way for, first of all, they need to be shielded. And one of the ways of shielding these lipids is by incorporating Hedgehog, inserting Hedgehog in the extracellular vesicles. But now, to go on on your question, Hedgehog doesn't display any activity, and mainly not the long-range activity, when it's depleted of this appendix. So it needs to be dually modified to be secreted and to display its role as a long-range morphogene, and even as a short-range morphogene in the wing imaginal disc.
So this hedgehog is already matured when they secrete through the EV. It's mature, it's not preformed. Preformed has no modification, I know. I would assume it's being already processed and to be functional,
Hedgehog doesn't reach the plasma membrane if it's not processed before. So Hedgehog is produced, then there is a signal peptide that is cleaved, and then Hedgehog has first an attack, cholesterol attack, that is going concomitantly cleave the C-terminal part and add the cholesterol on that. And on the N-terminal, you have a palmitoylation. I think that I have the figure here. It's not very to describe here, but you have the palmitate here and the cholesterol. And it's only under this form that hedgehogs travel to the apical and reaches the plasma membrane. So there is no way of incorporating hedgehog without the lipids in the extracellular vesicle. This was your question, right?
Yeah, I was thinking if the hedgehog is a free form or is associated with vesicle to travel. So there's I assume probably it's just one way or both ways they can travel to the downstream target cells.
Well, hedgehog cannot travel free because it has this dual modification, so it's highly hydrophobic and it cannot be released from the cells.
Gotcha.
So you have different ways of having hedgehog secreted. Now you have the extracellular vesicles, and also exosomes carry hedgehog, but in my hands, it appears that the exosomes are required for the short-range signaling, whereas the vesicles derived from microvilli are required for the long-range signaling. You also have, like, how can I say, a clusterization of hedgehog, and in that case, they bind to lipoproteins, and the lipid moieties are shielded, but otherwise hedgehog cannot signal. You really need, it's actually counterintuitive because you have these two lipid modification, which renders the molecule highly hydrophobic, but nevertheless hedgehog is secreted and act as a morphogene.
Thank you, thank you.
You are welcome.
Thank you, and maybe I'll carry on just a few more questions on this same question, on the same topic before we move to another question. So one question I had on your images of the hedgehog in the prominent knockout cells, it was intracellular, but in what compartment was it? But I guess from what you just said, it was probably in the endoplasmic reticulum or something like that. Did you check that? Yeah.
Yes, we did. So hedgehog reaches the apical, the subapical membrane. But it's in little, well, what happened is a little bit, probably in endosomes, because hedgehog, during the journey to be secreted, hedgehog has a very complicated route, and I did not want to show, but I can share that with you. So hedgehog from the trans-Golgi is, after being cleaved and everything, goes up to the apical membrane. This is what we call the primary secretion. Hedgehog is then re-internalized via dispatch, which is a cholesterol protein as well, associated protein, and it's internalized in a Rab5 endosomes. From there, it recycles back to the apical within the Rab4 endosomes. And once again at the apical, we observe by doing some proteomics and some genetic experiments that escort is also associated with this with hedgehog secreted in the extracellular medium. So the more likely is that hedgehog, when it reaches, I mean, it's close to this apical membrane, it's in endosomes. And we look at that. But
then when you knock out prominent, as you showed that it's not released anymore in the vesicles, but at what step of this pathway do you think prominent allows release of hedgehog? Just because it reaches the plasma?
Here. No, because the first secretion, hedgehog stays inside, goes to the apical membrane, but to the inner that's what I observed, that Prominint needs to go up here to the Hedgehog needs to go up here to the microvilli. However, Prominint and Hedgehog, they don't travel together to the microvilli.
Yeah, so there's still some questions there.
Yeah, and the question, I guess, is because this patch is tightly associated to Hedgehog, and this patch is also a cholesterol associating protein.
Okay, yeah, thank you. So there is another question from Sharif Ibrahim. Can you unmute yourself?
Yes, thank you. Thank you for this awesome presentation and nice work. I have a simple question about how you isolate or separate the large and small vesicles and how you define them. Because I'm doing something from 468 cells, doing something similar to that. So it would be helpful if.
Well, I mean, you mean for
the small and large, the MDA 468 cells, for cancer work.
MDA 46. Oh, by doing ultracentrifugation, because Drosophila is almost impossible to isolate from the hemolymph. No, it's just the current protocols that Clotilde published. I think that 10 or 15 years ago, 2006, right, Clotilde?
So you mean you have?
You mean okay.
To isolate large and small, you need to carry out ultracentrifugation, and what we do is that we keep the 10,000 G pellet.
10,000.
Okay,
10,000 and 100. Okay.
Right, but you have the whole description in this current. It's current opinion. What is the name?
Current biology.
Biology. Yeah, protocols in current biology or something like that, right? It's 2006. So go for that, and yes, the whole protocol.
Okay.
I tried this, but now I am trying to use the filtration, ultrafiltration system. So thank you so much for.
I mean, you are welcome, but check her protocol.
Okay, thank you. Thank you.
Okay. So there is a question from May Mahoney.
Okay, so I'm going to show my ignorance in this area, and I apologize for that because I don't really know the system you work with well. But under the conditions that you do your experiments, can you see the primary cilium, and do you see a relationship between the primary cilium and EV biogenesis?
Well, this is not a bad question at all. Prominint is also present in the primary cilium. The thing is that in my experiments, Drosophila do not have primary cilia. That's why I didn't check that out. But there is, I mean, it's one of the features of this CD123 Prominint. It's retaining cilia in microvilli and in protrusions. But unfortunately, I couldn't carry that out because the flies don't have any.
Thank you.
You are welcome.
And in fact, yeah, I had actually a similar question. It was based on one of the EVClub presentation we had a few weeks ago from the group of Maureen Barr, who was indeed analyzing the EVs coming from the primary cilium in C. elegans. So I was wondering whether you had a chance to compare, I mean, to see her data, whether there is Prominint also in her EVs. I didn't check that. And Hedgehog maybe or something, but I guess you did not.
No, I didn't check, but I mean, I am, you know, that I continue, I keep working on CD123, and so in mammalian cells now and in other systems, so I will probably end up by looking at the primary cilium.
And maybe another question on the biogenesis. So this ATP8B and quail inhibition that you use to inhibit the microvilli and in the Drosophila, do you know? I mean, if would you use them also in mammalian system? Would they work the same way? Do you think they would prevent the release?
I'm sure they will. Because I mean, quail is the vilin protein, is just the actin bond, is one of the actin bonding proteins. Is quail is in Drosophila is the name. However, ATP8B, there is a bench of ATP8B proteins. There is a huge family of ATP. I think there are 12 ATPB proteins in mammalian, and they are. I mean, they are known to flip In general, they flip phosphatidylserine. That's what is described. The thing is that we do not know what they flip in flies, if it's PS or not. But I think that people are starting to think about using these flipases. And I guess it's very interesting because this microvilli, and it's the same for the cilium, when you have prominent, you have these cholesterol-enriched domains, which are actually called cholesterol rafts. And by flipping this, breaking the lipid asymmetry of the plasma membrane, we might probably end up by affecting the EV secretion from this kind of protrusions.
Yeah, I think that's what Anne Weyman had done for the microvesicles from plasma membrane.
She was using TAT5, yeah.
Right, but it's a flipase. I mean, it's a
flipase as
yeah,
well. It's not the same one, but it's another kind of flipase. Yeah, she was. I think she published that in 2012 or 14. Yeah.
Okay.
It's interesting. I'm sorry. It's interesting how this hedgehog is, if this is in the it's encapsulated in the EV, how this orientation is, how this hedgehog will be presented to a target cell. It is buried inside the EV. For example, you have the one side is the formulation, the other side is the cholesterol, and I just couldn't imagine how this signal, because this is obviously is a ligand, act as a ligand to a receptor, right? So how this orientation is going to be turned around and then act as a ligand.
Well, my understanding of the question, you are asking me how these vesicles that carry prominent and hedgehog are able to signal.
Yeah, something like that. It's I mean,
okay. I believe that it's like hedgehog was showing to signal by binding to its patch receptor in the anterior cells.
But I think, yeah, I think for one question was also the membrane orientation of the hedgehog, because so is it inside or the vesicle or exposed outside? From what you say, you say that it's more exposed outside of the vesicle, right? Rather.
Well, I cannot. Yeah, I think it's, you know, I think that the cholesterol and palmitate are inside the vesicles and the head of hedgehog is outside. That's what I think. But it's difficult to imagine that this is only, I mean, it's not that it's difficult to imagine. I mean,
But if the, I mean, if hedgehog has a signal sequence, so it goes into the endoplasmic reticulum and then it's palmitoylated and cholesterol. So it means that this palmitoylation and cholesterol go into the membrane, but hedgehog itself remains in the lumen of the ER. And if so, it should remain external on the surface of EVs, because the lumen.
Not with the lipids.
But the lipids are inside the membrane. That's what inserts hedgehog in the membrane.
And
so
then the core of hedgehog is outside the membrane.
Is outside, yes. That's what. That
I'm sorry
would be
if I didn't explain.
Yeah. This is what I saw. That maybe your illustration maybe had to change a little bit, because if you look at that, it seems everything is inside the vesicle. Then
no. Where do you think that it's inside the vesicle?
No, the red, the green ball is outside.
Green bubble is sorry.
The green bubble.
Yeah.
Is outside.
I think that I put it outside, but outside, maybe you are right.
Yeah. Okay.
It's outside. Look.
Yeah. Okay. That makes more sense to me.
Maybe it's in the microvilli. The orientation in the microvilli is less clear, because we don't really know if it's.
This is something that I don't know, but just to let you know here, when you look at by immuno-EM, don't forget that when you do immuno-EM, you can see you cleave and you can see the whole thing. So this central dot here doesn't necessarily represent the inside of the vesicle. Could be the membrane that it's, you know what I mean?
Yeah. Okay, great. Thank you so much.
You are welcome.
So So, well, maybe I have a very short last question. In your mammalian MDA 468 cells, you had prominent both on the small and the large EVs.
Yes.
So you think that some of the small EVs could be exosomes, or you think the small EVs are only plasma membrane derived in this
No, I think that they could be exosomes.
You think they could also be exosomes?
I think so. I think so because you know also that breast cancer cells, they release a lot of extracellular vesicles. And I don't think it's a mix because, I mean, it's really well prepared and we observe, although I think that if I'm not wrong, is much less expressed in the small EVs.
No, it's
similar.
Well, it's similar. Yeah. But you know, the small EVs, they can also bud from the plasma membrane.
Yeah.
They are not necessarily exosomes.
They are not necessarily exosomes. I don't know, but this is something that is peculiar and very intriguing because, you know, we keep saying that prominent is also for microvilli. When we see that, we say, well, so now I'm not calling them anymore exosomes or ectosomes. They are small and large.
Yeah.
But this is something that we need to really analyze to see whether, I mean, it's inside the MVBs or not.
Yeah.
It was very difficult to do that with a wing imaginal disc because one cell is 3 microns, one single cell. So now with mammalian cells and the cell lines that we are using is much easier to look at that. Okay.
Okay. Well, thank you very much again, Gisela.
Thank you, Clotilde.
Great talk. So if there are no further questions from the audience, I think we can say thank you again and see you next week for the next week's EV Club.